Okay, so this is going to be the segment. Let's talk about the light microscope. The um, the light microscope, or LM for short, often is basically made up so that there is a light source and it's passed through the specimen. And when light goes through a specimen, um, the light kind of disperses, right? And you see kind of shadows. And when the light disperses, is basically called refraction. Okay, so it's basically the light is bending on the edges of, basically the main parts of the specimen. And this allows the specimen to be able to be seen under magnification. Now, there are three important parameters of a microscope. There's magnification, there's resolution, and there's contrast. Magnification is basically the ratio of an object's image, its size, right, to the object's image in the microscope to its real size. So the real size, of course, is smaller, right? The ratio is going to be whatever times more than that. So you can magnify an object, let's say 10 times more than what its actual size is. Now, the resolution is basically a measure, and now this is kind of going back to that super resolution microscope, of the clarity of an image. So it's a number or some measurement that represents the distance between, or the minimum distance that's actually visible to distinguish two things from being one thing. I'm going to talk about that. It might not make sense now, but I'm going to talk about that when we see some examples. And contrast is basically an idea in which there are differences between specimens, and sometimes you can't see those specimens. With a better contrast, right, you can see those differences, right? So we'll see some examples in just a moment. Now, let's talk about the microscope. There's several parts of a microscope, and I'm going to talk about first the arm right here. Okay, so you can see the arm extends out and it runs along the back, or sometimes called the spine, to the base. Okay, so the base is really what everything is sitting upon. It's the arm and the base that you must hold when you carry a microscope. So you grab the arm right here from the back, right, with one hand, and then the other hand you place underneath the base. You should always hold the microscope with two hands. This microscope here is relatively old, right? But they are still pretty expensive, and as you deal with newer microscopes, it gets even more and more expensive. So it's important to care for those. Now let's work our way from the top to the bottom. The ocular lens, or the eyepiece. So here, this is a binocular microscope. So that means there are two ocular lenses, one for each eye. You should be able to separate the distance further or closer together based on your what's called interpupillary distance, right? So you adjust it so that it is comfortable for you to be able to look through it using both eyes. I've seen so many students use only one eye. You should be able to comfortably, you set the distance, right? So that you can comfortably look with both eyes. No need to strain. Now, this is the binocular eyepiece tube. Okay, so this can be swiveled around so that you can have the eyepieces facing this way or this way. Let's see, we've got the revolving nose piece. Okay, so this is the nose piece. It revolves, and what's attached to it are the objective lenses. So we have two sets of lenses here. We have the ocular lenses and we have the objective lenses. Now, the ocular lenses are usually 10 times. So basically, they magnify an object at least usually just 10 times what its actual size is. The objective lenses magnify even further. So this objective lens says 4x on it. This one says 10x, and this says 40 times. So this is four times, right, magnification. This is 10 times, and this is 40 times. Now, in order to determine the total magnification, you must multiply the ocular lens times the objective lens. I will do a sample calculation in just a little bit. Now, I already talked about the arm, the mechanical stage. The stage is this whole flat piece in which your specimen sits on. Now, it's a mechanical stage because there are knobs here. So we have mechanical stage controls, which allow you to move your specimen or the slide, right? So normally it's a slide in which your specimen is on so that you don't have to move it with your hands. Okay, so you use that. And then also you just kind of lift this lever right here, okay? Or I guess push on this lever right here and it opens this up so that you put the slide in. You do not put the slide underneath this arm right here. Okay. The arm just kind of wraps around it like a hug. So it kind of holds it in place so that you can use the mechanical stage controls, right? So one moves the slide side to side, one of them moves back and forth. Okay, so you use the mechanical stage controls in order to move your specimen. Now, this is the illuminator right here. So this is your light source. Okay, so the light is going to turn on, right? So when you turn it on, the light comes through the specimen up through the lens, right? Now, in order to focus your specimen, you use these knobs right here. They're coarse adjustment knob and the fine adjustment knob. The coarse adjustment knob is coarse, meaning that the stage raises quite a bit easier. Okay, so the coarse adjustment knob is always used only when you are using the smallest objective lens magnification, and this is the 4x. It's called the scanning objective. Now, you do not use the coarse adjustment knob when you are magnifying it a lot higher because the stage will move too much and you're going to lose your specimen. You always use the fine adjustment to fine focus once you're up at the higher magnifications like 10x and 40x, and then 100x if the microscope has it. Now, I already talked about the base. Now here, what's not shown are the condenser with the iris diaphragm. These are actually kind of important. Unfortunately, you won't get a chance to actually use them. The condenser with the iris diaphragm are just underneath the stage. So they're underneath there. The iris diaphragm allows you to open and close the amount of light, so reduce or increase the amount of light that goes through your specimen, which can increase your contrast and resolution. Okay, so that can help improve that. All right, so those are the main parts of the microscope. You'll get a chance to review those in the microscope lab. All right, so light microscopes can, like I mentioned, you could have a 100x objective, but this allows you to magnify up to 1,000 times the size of the actual specimen. Now, there are a number of different techniques which can increase, so that's magnification, but there are a number of techniques which allow you to increase the contrast of a specimen. And staining is one of them, and labeling with fluorescent molecules is another. Now, the resolution is usually too small for a light microscope to study the organelles, so you have to use the super resolution microscopy in order to study the individual organelles that are inside a cell. Now, let's look at some different types of microscopes. Now, this shows a light microscope here, and this basically compares the difference between an unstained specimen and a stained specimen. So you can see the contrast is greatly improved with a stained specimen versus an unstained. You can imagine this would be very difficult to find if you were looking at this. Looks like a representative animal cell. Often the cheek cells are used. But when you stain it, you can easily see, and you can kind of see they're overlapping right here. You can see the nucleus a lot better than you can see it over here. Now, these represent some of the organelles inside there, but you can't really tell what each of the organelles are within these cells. Now, phase contrast is a type of light microscopy or light microscope, which allows you to see a lot more depth and not really three-dimensional, but you can definitely see the depth a little bit better. So you can see this overlapping, and you can see that it's a lot the cell depth. You can kind of see that with this type of microscope. Now, here, differential interference contrast, or Narmarsky, which is it's commonly called microscopy, allows you to see more three-dimensional type depth, of course, but it looks more 3D. So you can see the nucleus. Here, the cell looks a lot more flat. However, the nucleus does not. It looks like it projects out of the cell, and you can see actually where this cell is underneath this cell, and it's lifting up the top cell right along the edges. So you can see that a lot better, more three-dimensional. Okay, so now we're getting into some more exciting bright colors. This is fluorescence microscopy. And this kind of shows a representative example of fluorescence microscopy. However, we're going to see some other examples which uh, this may actually be because it's pretty clear. However, the point of fluorescence microscopy is to allow us to actually label molecules that are inside the cell with different colors, right? Really, they're just labeled with a molecule that fluoresces under UV light, which is the source of light in these types of microscopes, a specific color. So here we can see three different colors, right? So we've got the blue, we've got the orange, and we've got the green. The blue, of course, is the nucleus. The orange represents some of the organelles like the mitochondrion, and the green represents part of the cytoskeleton of the cell. So you can kind of highlight different structures within the cell, and this allows you to see a live cell moving around, right? So you can see the things moving inside the cell. Confocal or deconvolution microscopy allows us to see a lot more detail, right? So there's special software that's actually used to be able to make this what might be considered out of focus a lot more, let's say, in focus, right? So, and it gives us a much sharper image, and it's three-dimensional as well. Now, confocal microscopy. Here we can see standard microscopy, right? And then we have the confocal microscopy, which is quite interesting. It actually improves that what might be considered out of focus with standard microscopy. It improves it to be a lot more in focus and a lot clearer, so you can see much more detail. Again, this is kind of three-dimensional as well. Have a super resolution microscopy, and this shows basically standard microscopy, and then this is super resolution. So remember, resolution is basically the distance with which you can distinguish between two items or two things, part of your specimen. So here, this looks like one thing right here. This looks like one, this looks like one. But then when you have super resolution, you can actually see that it is not one thing, right? So they're so close together that it appears as one thing when you don't have a really good resolution. Now, when you do have a much better resolution, like super resolution microscopy would, you can see that there are individual parts of this specimen. So you can distinguish between two very close parts of the specimen. Uh, Electron microscopes are quite interesting. They provide a lot more, a better way to see some detail, right? So we have what's described in your textbook is scanning electron microscope and the transmission electron microscope. So when you see a figure inside in your textbook, you're going to see them all throughout the textbook. It might say SEM or TEM. Okay, so that means this image is from a scanning electron microscope, and it means TEM would mean that it came from a transmission electron microscope. Now, scanning allows you to see the surface of things. So here we can see on the surface of the cell, we can see the cilia. Now, this gives us a three-dimensional view as well. Now, as opposed to scanning electron microscope, the transmission electron microscope lets us be able to see within specimens. So it allows us to see within, here in this case, the cilia. So we can see longitudinal sections of the cilia. Okay, so that's along the length of the cilia. And then we can see cross sections of the cilia. So if you've already read this chapter, you're going to notice that, remember, the cilia are made up of microtubules along the length of it. And when you cut a cross section, you might see this 9+2 arrangement of microtubules. All right, so in your text, it might mention also cryo-electron microscope, right? So this is basically tissues where they were frozen rapidly, and allows us to visualize by electron microscope some details of smaller objects like a specific protein. Okay, in summary, I talked about the characteristics of a microscope, the three important parameters, the microscope parts and what their general functions are, and also all the different types of microscopes. Now I'm going to do a sample calculation in a separate file, so look out for that. Thank you.